very big eye. So the idea is the following. You have a single eye field, okay, which is PAX6 positive. Sonic comes in the midline because it is a midline factor. This is the importance of the midline in the patterning of the body. Midline is a value in development, okay? It's not only a value, it's a barrier. It's a barrier. I can discuss that with you later. So sonic comes in the midline. Therefore, there is no PAX here and no eye development. This is, guys, why we have two eyes. בסדר? Why we have two teeth, right? So sonic hedgehog inhibits PAX, and then we have two, and therefore we have two eyes. So let's do the same exercises we did before. So smoothened for us is downstream sonic activity, right? So smoothened as sonic, in this case, will inhibit the central field, and we will have two eyes. What happens if we inhibit smoothened? We will have one eye. We will have a cyclop, right? What happens if we inhibit patched? Of course, we will have two eyes because we inhibit the activity of smoothened. In this case, it's the opposite as with the spinal cord, because sonic plays not an inductive role, but an inhibitory role. What happens if we inhibit patched and there is no sonic? So there is no smoothened. What happens if there is no patched? Perfect. So we know now what happens. What happens if there is no smoothened activity, but there is patched and sonic in the system? Of course. So why do we call the cyclop cyclop? And this is where we are going to finish. Because there is a drug coming from a plant which is called the Veratrum californicum. And this drug that we use in the lab, it is an extremely potent drug. It's an alkaloid, a little alkaloid. It is called cyclopamine. Now, when we apply cyclopamine, we basically inhibit the activity of sonic. What does cyclopamine do? It's a potent inhibitor of smoothened. It binds to smoothened, and it inhibits the activity of the smoothened co-receptor, thereby inhibiting the activity of sonic. If it inhibits the activity of sonic, you know that you are going to get a one-eye field instead of two. This is why we called a cyclop a cyclop. Set up? Okay. So let's make a break here. Funny. Okay. Questions?
When you just have one eye, can child to see?
I think they can see, but since this is not the only phenotype, these youngsters, because we have cyclops among animals too, they cannot survive for long. I don't think they have ever been tested for visual activity. I don't think they have ever been tested. We see it sometimes with our chickens. With our embryos, we see sometimes double axis, so they have two spinal cords and then one eye in each side. But this is it. They cannot survive because never cyclops come on their own. There is a lot of other brain malformations which are incompatible with life. You can create a cyclop. It's not difficult. It's really not difficult. But, I mean, everything in the brain is completely distorted. So what are you going to ask? How are the pathways created? How is the normal innervation? There is no optic tectum. There is no bifurcation. There is no contralateral and ipsilateral innervation. Everything is completely mispatterned. So, and it's not the only thing. It's not that you can create only, basically you could create only a cyclop by inhibiting sonic activity only in the eye field. We can now inhibit sonic activity, maybe under the regulation of the PAX promoter. That would be a possibility. I don't know if anybody did it. And then you see, try to. But PAX6, no. You're going to get also problems because PAX6 is not only expressed in the eye field, it's also expressed widely throughout the neural tube. So you're going to get all kinds of mispatterning problems. Other questions? So let's have a 5-minute break, 5-10 minutes.
Just one small question.
Yes.
Is the cyclop named after the cyclopamine, or is the cyclopamine named after the cyclop? Because cyclops appear in Greek mythology, right? So
Yeah.
I
I guess this was the idea. And I don't know which comes first. I don't know which comes first, as a matter of fact. I don't know whether the cyclops were called cyclops at that time. I mean, the cyclops existed forever. You have them in mythology, as you say. But were they called cyclops already? Look at it and tell me. Look at it and tell me. Email me. I never looked at that issue, as a matter of fact. It's called cyclop forever?
Yeah, because it means round eye.
Yeah, maybe. Maybe. So this is why they called the drug cyclopamine. Yeah, because when you apply it, you can cause, yeah, that makes sense in my view.
I had a quick question.
Yes.
How often, when sonic hedgehog is disrupted, how often do you get something like cyclops? Can you control it, or is it 50% of
No.
these experiments?
No, it's not penetrant. I don't know exactly. I don't know exactly what is the percentage, but not in all of them.
Okay.
Not in all of them, certainly not.
Yeah.
Because all these mutations are not that penetrant. The sonic pathway is so complicated, and as I told you, it appears there are non-canonical ways for activity of sonic as well, which we only begin to understand now. So it's the tip of the iceberg. So I guess that it will take us still a lot of time to try and understand what's going on there. Yeah, it's not fully penetrant. It's not that every time you have a sonic mutation, you will have a cyclop. Not at all.
And what about the cyclopamine, the drug?
Yeah.
Is it stumbled by accident, or
I don't know.
how do they associate this plant, which seems quite random?
I don't know. It's like many plants that give us a lot of clues to things. I have no idea, as a matter of fact. But Google it and tell me. This is why I was told here that the word cyclop existed for a long time. So it appears that this was the first. So the cyclop existed first, and then the plant, because it was known that it inhibits sonic and gives rise to generate cyclop, this is how it was called or termed cyclopamine. This is what I think. But I asked them also to Google it. I don't know what comes first, as a matter of fact. feels in the peripheral field.
Okay.
Yeah, because tonic is expressed there. As I mentioned, tonic is expressed in the gonal polarizing activities. And then if something in the gradient, it's not necessarily tonic, there is another molecule called the GS, okay, but if the interaction between the two, something goes wrong, this is what basically you can get. It's quite common, by the way.
Yeah, I've met so many people.
Yeah. Never mind.
Call me a frog.
Never mind. You are okay.
That's good to know.
We are all screwed up some way, believe me. None of us is perfect. אז תוכל לשים להם את זה.
רציתי רק לבקש לך משהו אחד שנשמע לי קצת, לא הבנתי אותו, מה שאמרת. הטוניק, אמרת שהוא... טוניק, אמרת שעובד על הפרומוטור שלך, שהוא נכנס לגלים.
הוא לא עובד על הפרומוטור, הוא קושט, הוא נקשר.
אבל אז אומר שהוא צריך להיכנס לגלים.
זה לא טוניק.
זה לא טוניק, זה דרך הגלי, אוקיי.
זה הגלי,
חשבתי שהטוניק עצמו.
לא. טוניק מפעיל את גלי, גלי נכנס לגרעין, גלי נקשר לפרומוטור של פאץ' ואז הוא מפעיל את הסינטוז שלו.
לא, אני חושב שאני אולי התבלבלתי, אבל מה ש... כי יש משהו קצת מוזר בזה שאתם כאילו מדברים על סוניק אקטיביטי, שאת עושה איניביציה על סמוזינג. ואז סוניק בעצם הופך להיות אי-רלוונט, כי הוא בעצם זה דירקט. אז זה קצת מבלבל, כי זה כאילו מה היה קורה במצב הנורמלי.
למה זה לא עולה?
מה, אתה רוצה שזה תאמר אותו?
משהו
לעשות update מהאופרטים.
אני רואה את ה...
לא, אני יודע.
חכה, אולי לא שמתי אותו במקום אחד.
לא, אני חושב יש לי עוד אחד, אבל אני רוצה לצאת את זה.
נתאזר בסבלנות.
כן, שום דבר. הנה.
בבקשה, אז זה 4 אלסק ניורל קרסט DNS. רק תוכל להזיז, שוב פעם אני לא
אחרי זה שואלים אותי אם הילד אופטימלי, זה כל מה שרציתי לדעת.
הנה, כבר עבר.
כן, אילן קבע לי איזה משהו, בלאגן שהעסק הזה. סתם עם כל הרביטים שלו, זה מפחיד. מה?
בגלל שצריך להשלים, אני אמרתי לליבי, צריך להשלים את הכל לאותו מחשב, ושכל אחד שבא לדבר,
בקומיטי.
לא כולם לא יכולים לדבר, יש להם...
לא, אבל בעניין הזה שכאילו שולחים את המצגת. לדעתי הם עוד לא החליטו בכלל מי מציג ומי לא.
לא, אני חושבת שכן. לפחות אנחנו ביקש ממני, ממוני ומאילן.
כזה?
מה? לא יודעת. זה החלק של המולקולר, משהו, כאילו שאנחנו מלא קבוצות, אתה מבין?
גלעד שנייה. Just before we start, some of you were not here on the first meeting, so I want to pass this page again. And the ones that do not appear on the page, please add yourself so we can have the whole list of people that are participating. Okay, and please write whether you take it for credit or not, because this will affect whether you are presenting a paper here somewhere.
What is this lateralization? מה קרה פה? It's because you came from... Think about it. Okay, so as promised, this mini course that I'm giving you is actually a taste of developmental neurobiology. So it's not that we have nothing to say about the central nervous system any longer. As you may imagine, this is the basis of a course. Whoever is interested in the faculty, I don't know if this year or not, but we run every 2 years a course in selected topics in developmental neurobiology, a course based on seminars and a lot of interaction between students that may interest you in the NCAREM campus. So this brings really questions of phase specification, axonal guidance in aptogenesis, pruning, etc., etc., etc. Most of the relevant topics of developmental neurobiology at quite a high level. So if this is of interest to you, this may be a possibility in the second semester. I don't know if your program is closed or you may take courses from outside as well. So don't forget we have a campus in the hospital as well, next to the hospital, in which a lot of neurobiology is being done. So after this publicity, what I would like to go with you through now is a little bit on the ontogeny of the peripheral nervous system, which is a very important branch of the nervous system because it is composed, as you will see in a few minutes, of all the sensing machinery of your body, which transmits signals to the brain and which also passes signals to your viscera to enable you to function. And I will give you a little bit of evolutionary perspective on that and show you some, I think, interesting things about that. Okay? So let's go back to what we studied last week. And this is, again, the process of neurulation, the formation of the nervous system. Ectoderm, neuroectoderm, ectoderm again. And I would like now to concentrate on this area, which is now here having these stippled lines in white. This area, which is at the junction between the ectoderm and the neuroectoderm, which is a continuum, okay, will form what we call the neural crest. So once the folds come together and they close, and this is now a transverse section through the neural tube, what you see is that we have a very particular set of cells that morphological-wise are or look exactly the same as the other cells of the neural tube. They are a pseudostratified epithelium. We learned this term also last week, namely one single cell spanning the entire apicobasal extent of the epithelium. But molecularly speaking, these cells are already different. For example, what you see here is the in situ hybridization or the expression of an RNA coding for a certain transcription factor, in this case SNAIL2, doesn't matter for you, but this is what makes this area of the neural tube very different in terms of its fate when compared to the rest of the CNS. What is different here? That these cells will basically leave the neural tube, they will leave the neural tube, not become integrated into the central nervous system, but they will leave by a process of epithelial to mesenchymal transition, EMT, that we studied in the laboratory for over 10 years, and discovered or uncovered the molecular network that operates to produce this emigration of the cells, and then these cells will migrate throughout the embryo to produce a lot of derivatives. So what is different from CNS and neural crest is that, distinct from the CNS progenitors, crest cells are going to leave and to migrate throughout the embryo. They will do it through particular stereotypic pathways. All of them have been mapped in our laboratory, in many laboratories in the world. We know when the cells emigrate, where the cells migrate to, and when do they stop migrating to form peripheral ganglia, etc., etc., what you're going to see now. And what we are obviously learning now, among other things, is how do these cells acquire their final identities, and they are many, as you will see in a minute. How do they migrate molecularly speaking, namely which are the guidance molecules that operate to tell the cell, do migrate through this pathway, but do not migrate through the other pathway, and stop migrating now, and not before and not after. Otherwise, again, many malformations will take place, and there will be no coordination between peripheral and central nervous systems. So this system is of utmost importance in the embryo. So this is how the neural crest looks after it has emigrated from the tube at the cranial level, in your head. This is how it looks, very differently from what it looks in the trunk, because in the trunk, the neural tube is more elongated and the cells are more discrete when they leave the tube. In the head, they will live as a group of cells. In the trunk, they will live virtually one by one, throughout many days until the process of emigration is completed. Now, why is the neural crest so important? Why is it so important? Because it will generate most, not all of the peripheral nervous system. I don't know if I will get into it, but not all of the peripheral nervous system, most of it. And it will also generate, this was a nice idea, but I have to use it. It will also, this is nice. It will also generate many non-neural derivatives of your body, because the neural crest is evolutionarily a very important system which arose only in vertebrate embryos. Non-vertebrates have no neural crest per se. Have certain cells that people play around with and claim they may be neural crest progenitors, but truly it's an invention of the vertebrate system, and we'll get into it in a minute. So let me go very briefly through this list. It's not an exhaustive list. You don't have to know it by heart, but it's an important list. You will see why. So first of all, among those derivatives of the peripheral nervous system, sensory ganglia. You know that we have sensory ganglia along the entire spinal cord. These are called the dorsal root ganglia. They have many different types of neurons which transmit pain, temperature, touch, proprioception. If anybody doesn't understand the term, please ask. Proprioception, which is the sense of where you are in space, and so on. Each of them, of these senses, is transmitted by a different subset of neurons. And in the dorsal root ganglia, all the kinds of neurons, as well as glia, satellite glial cells, arise from the neural crest. Okay? Then, so you have spinal ganglia. These are the dorsal root ganglia. But you also have all kinds of cranial sensory ganglia. So in your head, there are many sensory ganglia of the trigeminal, of the glossopharyngeal, the facial nerve, all of the nerves, the 7 nerves, the 7 paired nerves that leave your brain, okay, have also a sensory component. These are the cranial sensory ganglia. In these cranial sensory ganglia, all the satellite glia derive from the crest, and in some of them, some of the neurons come from the crest, and in others, the neurons come from a different source, which is called the placodal ectoderm. The placodal ectoderm is a thickening of the ectoderm, of the skin, which will generate neurons that will then join the ganglion and form different neuronal cell types. Doesn't matter. This is, again, a couple of hours to give you a speech. This is just to justify why I said that most of the peripheral nervous system, but not all of it, comes from the crest. Since in cranial sensory ganglia, some sensory neurons arise from the placodes and not from the crest. This is all. All the rest comes from the crest exclusively. So, cranial sensory ganglia, many components come from the crest. Then, satellite cells in all the sensory ganglia, as well as autonomic ganglia, all the satellite cells are crest derivatives. Autonomic nervous system. This is a very interesting system. The autonomic nervous system is formed or composed of 3 branches. The sympathetic system, which is obviously a motor system, and is organized into sympathetic ganglia, sitting next to your notochord or your vertebra. The parasympathetic ganglia, which actually sit in the organ of homing, be it the head, be it, for example, the ciliary ganglion of the eye, or be it your gut, in which the parasympathetic ganglia sit within the gut itself, and then modulate intrinsic enteric innervation. And the third branch, which is a very important one, what I call the second brain, is the innervation of your intestines, of your entire digestive system. This is also coming from the neural crest. Now, the innervation of the gut, again, a talk in itself, arises from two levels of the crest, from crest at the level of your, what we call the vagal level, the level of your neck, and the sacral level of the body, and cells migrate to the gut mesoderm, and then once they arrive there, to the esophagus area, they migrate all along to innervate, to create neurons and glia that will form two plexuses, because you have in your gut networks of neurons. If anybody has seen that, and they are two networks, the Auerbach plexus and the Meissner plexus, and they are those which are responsible for peristalsis, namely for the movement of your bowel, for the contraction of your stomach, and so on and so forth. And then these get an innervation of the sympathetic and parasympathetic system, which modulate the rhythmicity of peristalsis. Namely, if we want to have slow contractions of your bowel or very fast contractions. But if we do an immunosympathectomy, if we cut the sympathetic innervation, your bowel for a while will still contract and undergo peristalsis and be able to move your food. Why? Because the innervation of the enteric nervous system is an internal innervation, also produced by Crest. So many people will call it a system in itself. So complicated. The number of neurons and glial cells in the enteric nervous system is approximately that of your spinal cord. So we are talking about very discrete number of initial cells, of Crest cells, that lead through proliferation and migration to an enormous repertoire of neurons and glia of all kinds that will localize to many parts of your body. So, autonomic nervous system, the sympathetic, parasympathetic, and enteric nervous systems. Then, all Schwann cells of the peripheral nerves. As you know, myelin is formed by Schwann cells, and this will produce the saltatory mechanism of nerve impulse transmission. Now, in the peripheral nervous system, the cells responsible for ensheathing your nerves are Schwann cells, and they are all derived from the crest. In the central nervous system, the cells ensheathing nerves are oligodendrocytes. So there is a difference between the origin of the cells that produce myelin in the CNS versus the PNS. In the PNS, these are Crest derivatives, the Schwann cells. Next, endocrine and paraendocrine cells. I'm not going to enter into this field. It's a very interesting field. Many endocrine glands in our body, thyroid, the adrenal gland, come from the crest. And these have a lot of importance in regulation of body functions. And then something which is very interesting. For reasons that we don't understand, all the pigment of your body, all the beautiful colors of the birds and of fish, and all these fantastic patterns come from the neural crest. This navy that we have here, all the pigment, okay, comes from the crest, except for the pigment of the eye. The retinal pigment, epithelium, comes from the diencephalon, directly from an outgrowth of your hemispheres of the brain, not from the crest. But all the pigment of our body comes from the crest. And this is very important because it appears, also based on our own data, that there are common progenitors for the generation of neurons and for the generation of pigment cells, which at a certain time in development segregate from each other. And there are a lot of diseases which involve Schwann cells, for example, and pigment cells. For example, people which have a mutation in the neurofibromatosis gene, in the neurofibromin gene 1 or 2, get a disease called neurofibromatosis, in which they get schwannomas. They get tumors of Schwann cells ensheathing the nerves. And these tumors are pigmented. or get pigmentation to different extents. Children which have Hirschsprung's disease, it's a disease of the enteric nervous system in which the colon, the distal part of your gut, does not get innervated, and the children cannot defecate. And the intestine becomes a balloon because it doesn't have contractility. These children have pigment defects. They have patches of depigmentation in the body or of strong pigmentation because the molecules that generate the autonomic nervous system also generate pigment, and so on and so forth. So this is a very, very interesting derivative of the neural crest. Many laboratories are studying this because it's also obviously the basis of melanomas, okay, of all the skin cancers. And then, last but not least, and this is where evolution comes into play, we have mesectodermal derivatives that come from the crest. What is mesectoderm? Ectoderm generates nervous system. Mesoderm generates muscle and bone. What is this chimera called mesectoderm? Now, it appears that in the body, all the bones of our body come from mesoderm up to this area. From here, from your neck, the base of your skull, to your tail. Why? You know that. You know that the notochord secretes sonic, which acts upon the mesoderm to produce your vertebra. I taught you this half an hour ago. But it appears that there is no notochord in your head. The neural tube at cranial areas has no notochord underneath. So the mesoderm of the head, which is called the cranial mesoderm, is for some reason unable to generate bony structures. At the same time, there was in evolution the process of cephalization, namely the process by which, as I also told you last week, a brain began developing, which is a specialization of the tube, which is much larger, which generates hemispheres, and also needs protection. Also needs protection by bone. Now, since the mesoderm cannot create this bone at cranial levels of the axis because there is no notochord and no sonic in that area that affects the mesoderm, there is a theory, and this is a theory, which was created by two guys called Gans and Northcutt. You will see it in the slide, and I gave the presentation to Mickey, who will pass on to you the presentation so that you have it. There is a theory which claims that with evolution and the process of cephalization and the need to have a protection for the growing brain, this brain that with evolution begins to be more and more prominent and concentrates all our senses, or most of our senses, and the transition in evolution from filter feeding to predation, namely these animals that now predate and need, all these senses to do it, all of this leading to the process of cephalization in evolution, the neural crest took over a function which it did not have previously. And this function is the ability to generate skeleton. Now, it appears that all of the skeleton of our head is from the neural crest. This that I'm touching here, my mandible, my maxilla, okay? And many even ossicles, little bones in my ear, come from the crest. So this is not a mesodermal derivative, which would be the normal thing. This is an ectodermal derivative, but a specialization of the ectoderm that we called mesectoderm. So it's very funny, it's very nice. And I'm not going to go through the list, but the list of bones, connective tissue, adipose tissue that derives from the crest in these areas is quite dramatic. So if we look here, what you see is that in red all comes from the neural crest. So this would be the skull of a chick, and all this comes from the crest, whereas the occipital bones come from the somites and from other parts of the cranial mesoderm. How would this look like in a human being? All these bones, including the maxilla and mandibula, come from the crest, whereas the basi-occiput and another occipital bone come either from the somites or from the cranial mesenchyme. So a very significant part of our cranium comes, in fact, from a structure which is an ectodermal structure and which is meant to be a neural structure. So the neural crest has a lot of evolutionary perspective and significance, and for whoever wants to read a very interesting yet old article, still very significant, go to Science and look at this beautiful article which basically presents a new hypothesis which is called the new head. And the idea of the new head is that we are basically two different organisms. We are a trunk and we are a head. Believe me, the behavior, I have like 20 years of experience with this structure, and I can fully confirm that the behavior of the crest and of many things in the head is completely different from that in the trunk. In terms of the trunk, we are more similar to fish. In terms of how the neural tube forms, there are things I did not tell you, obviously, of how we form a neural tube in the tail area of our body. It's not formed like that by invagination. It forms by cavitation, like in fish. So it seems that the more we go to the tail, the more we resemble an ancient species. The more we go to the head, the more we are new. So this is the new head hypothesis, and I'll be happy to discuss that another time with you. It's very, very interesting. Next, so the idea of the new head, Ken.
Fish are also vertebrates, right?
Yeah, but they are lower vertebrates compared to us.
So the neural crest is not developed the same?
Oh, yes, absolutely. The neural crest is very well developed in the fish, but in the fish the difference is that the ganglion, in our case, consists of, 30,000 cells, neurons, maybe more. And in the fish, a DRG is 3 neurons. 3. So each cell knows from the beginning what it is going to do exactly. Okay, a fish has also a head, as you know, with a lot of mesectoderm, which comes also from the neural crest. But it's much more underdeveloped than all our peripheral nervous system, obviously. And the way the neurulation takes place is different. It's not formed by, as I said, by invagination, but it cavitates. So you have a mass of cells, then you have apoptosis, cell death, and then a tube is formed. Okay, this is called the neural keel. And our caudal part of the spinal cord, from the umbilicus up to the tail, is formed the same way, even in human beings. And then it's formed here up to the brain by invagination. This is what we call primary neurulation, and this is called secondary neurulation. So here we are very much like fish and others, okay? And here we are like ourselves. Okay.
Are there any different cells to the neurocrist in non-vertebrate features?
Whether they are crest-like cells in non-vertebrates? Yeah, there is this very ancient vertebrate, which is the Amphioxus, which has some cells that are considered to be crest. And last week in a meeting, I met a guy from Berkeley who claims that in Siona intestinalis is the worm that sits in your bowel. It's an ugly worm, okay? Certainly not a vertebrate, right? And he claims that this ugly worm has pigment cells, and that when adding a transcription factor that we uncovered in the lab to monitor and switch between pigment and neural fate, when he adds to this worm FOXD3, this transcription factor, he is able to turn this melanocyte, this pigment cell, into a neural cell. And thereby he claims that although these melanocytes do not migrate like they migrate in vertebrates, because in vertebrates melanocytes migrate like crazy, these cells, although they are unable to migrate, they are still able to undergo a switch between melanocytes and neurons, thereby resembling an ancient neural crest-like phenotype. So there are people who play with the concept that ancestors of the neural crest already exist in non-vertebrate animals. But everything depends on what you define as a neural crest. A neural crest is defined as a cell which undergoes EMT, epithelial to mesenchymal transition, migrates to the periphery and generates components of the peripheral nervous system or the ectomesenchyme. If this is your definition of a crest, then the Siona and others do not have crest. However, different species have melanocytes, even worms. They have pigment cells. So is this crest or is this not crest? It's a matter of discussion. Okay, so this is under discussion. People like playing around with the concept. In my view, it's more of an intellectual type of game and a matter of definition. I am ready to call it neural crest-like, certainly not neural crest. Okay? Okay, let's continue. When should we... אנחנו לגמור. מתי? רבע, זה עברנו את הרבע. Let me see. Let me just leave you with a few questions, okay? Let me take just 3-4 minutes, because otherwise I would like at least to leave you with the open questions. So, why is the neural crest so sexy? Why is the neural crest cell such an interesting system? It's because it generates a huge variability of phenotypes, a lot of cell types. And everything begins with an extremely discrete group of cells. So the questions that people ask in their laboratories, and the questions which are of absolute importance, is, are crest cells multipotent? Namely, when we are all multipotent at a certain stage in development, the question is when and how do we get restricted to generate specific fates? This was our discussion from last time. Are crest cells, contrary to that idea, fully specified? Or is the crest a mixture of fully specified and multipotent progenitors? And basically, let me give you the answer. This turns to be the correct answer, but in contrast to the basic and classical dogma that crest cells migrate as multipotent cells, namely each cell is able to generate the full repertoire that I showed to you, we know and then they decide what to become when they come to their homing sites due to environmental signals. We know now that this is not true. We know that the decision to become neural versus melanocyte, Schwann cell versus pigment, etc., is already a decision that is taken place sometimes even before leaving the neural tube. Namely, very early during the creation of this cell type as an autonomous cell type different from the CNS. And this means, why is this important? This means that intrinsic factors playing within the neural crest, namely a transcriptome and microRNAs and other regulators, already enter into the game very early in development, creating a more deterministic type of mechanism for cell fate decisions, rather than a fully stochastic mechanism which brings every cell to every place, and then only when reaching the ganglion, local signals will tell the cells, do become this, do become that, but repress that phenotype. This is not the case. So this is just leaving you with a take-home message without demonstrating basically anything. So don't take my word for it. Just either go to the literature or come to my lab and I can continue elaborating on this idea. So I will leave you just with the last data slide that proves that crest cells are not multipotent. And this is based on some work a student of mine has done like 2 years ago, by which he developed a system in which he is able to follow the fate of a single cell in vivo, in the embryo. So what he does is he injects into single cells, never mind how, a DNA that encodes for GFP, for green fluorescent protein, which is a fluorescent tag. And you see here how he's able to just impale a single cell. These are cells which are 5 microns thick. It's not that easy. These are epithelial cells. And then he does this exercise at different stages, and then fixes the embryos, let's say 5 days later, and sees what is the fate of the cell that has been impaled at different times. And what he finds is the following. If he does this exercise at 25 somites, when the embryo has only 25 somites, always at the very same axial level, but at different stages, the cells go to the sympathetic ganglion. This is the sympathetic ganglion next to the aorta, and will form sympathetic neurons. If he does it just 5 hours later, this is the interval here, the cells go to the nerve and will generate Schwann cells. I'm not showing you all the demonstrations because time is short. If he does it at 30 somites, the cells will go to the dorsal root ganglion and generate sensory neurons and glia. If he comes a little bit later, the cells will go to the melanocytic area, namely to the skin, because they migrate differently. The melanocytes go only to the skin. And if he impales the cell at 40 somites, no more crest is created. The crest period has ended, and the cell will become a roof plate cell. The very same as we have a floor plate in the basis of the spinal cord, you have a roof plate in the upper part of the spinal cord. The roof plate is an organizing center for interneurons, etc. It doesn't matter now. But the idea here is that it's not that a single cell at any time is able to generate everything. What would have been the alternative? That a single cell at any time would have generated everywhere derivatives, in the DRG, in the melanocytes, in the Schwann cells, but they do not. This means, and we have found also the way the cells leave the neural tube and do so, and we have a molecular network that we are now studying further, etc., etc. So our claim is that at least in the trunk, there is a very early decision process by which cells, via an intrinsic mechanism and changing neighbors due to progressive emigration, are able to know where to go and what to become at a very early stage. So this is the message I would like to convey you. Google neural crest, go to papers, and you can a little bit enlarge your knowledge on that topic. Any questions? תודה. איך סוגרים את הדבר הזה?
אירופאית? כשאתם עושים את הסינגל הזה, כאילו לא לעשות את זה שייקספיר אלא משהו אחר?
פה אנחנו לא עושים את זה כל כך ברמה הקלונלית, אבל סטודנט אחד שלי אחר עשיתי לשנות ברמה קלונלית, וזה עושה דברים יפים. כי מה היופי? לעומת הדברים, מה שאתה עושה בצ'יפ, זה שאתה יכול להתערב בפייט של קרבוידט כשהסביבה נשארת נורמלית. וזה היופי. כן? ועוד שבאחרים שאתה עושה נוקאוט בטרנסגן, אתה דופק את כל המסה, והסל-סל אינטראקשן משתנים לגמרי. אז לפעמים האינטרפרטציה היא לא... כן, היא בעייתית. אז זה היופי של הדבר הזה. טוב, אני רוצה לך רק לבקש חופשי. מיקי תודה.
תודה לך. אני הייתי לך בקשר פעם, אני רואה את
שוב,
התמונה
כמו שהיינו
הזאת.
מגדירות את כל
הוא רואה
קאפלינג
אותו
בשביל למצוא איזה דקה.
כן, אותו
לעשות
דבר,
קצת
בדיוק.
לראות,
אז הוא מסתכל והוא אומר שמה כאילו, הוא אומר משהו על אשתו והדודה או משהו כזה, women are communicating in ultrasonic voice. You think you can understand what they say, but actually they're like dolphins.
אתה עוד שנייה
אבי זה אותו דבר.
שאני פולניה.
טוב, אז מה, אתה מתחיל? יאללה, בוא נסיים את
נתתי הרצאה, בשנה שעברה נתתי הרצאה יש קורס מותאם כל המחקר לפיזיקאים, שנה ג', אני מספר כל שבוע בעצם על כל מחקר המחקר שלי. הגעתי בפעם הראשונה לעולם של הארצות, לעולם עם מדרגות כאלה, זה שם באמת ממש מעלות, ואני פתאום איחרתי, הלכתי, ונתתי כניסה מרשימה, נתתי בהתהפכות על המדרגות.
מה, בדעת קריקס?
אני ישבתי פעם אצל איזה אורתופד, היה לי משהו וגם שלחתי לצילום, וחזרתי אליו לזה, והוא לקח את האקס-ריי, יושב ככה, כאילו, לכיסא לראות את זה מול האור, ופשוט אמרתי לו, זה יש לי בעיות גם חולים עליך.
אמרת שאנחנו מחכים למישהו, לא?
יש לנו איזה דף כזה שאפשר לבדוק את זה.
עכשיו הם קיבלו את האתגר שלהם?
לא, אני לא חושב שאפשר. וואו.
מה זה?
אני לא
זה
חושב
מפלט לי?
שאפשר.
לא, זה מפחיד. מה עושים את זה?
איפה הבאת את זה?
עכשיו התחננתי לו, היא אמרה זה רק סטודנט, אמרתי לה למה יורם קיבל?
אנחנו צריכים כיתה ליום ראשון, הלו, מה זה היה? יש לי אצבע שאני כולם שבליד כולם פה זה קשה לי. לא, זה לא בסדר. אני הבנתי, השנה הזאת עשית לי הרבה סטודנטים.
כן, אני חושב שאת הבנת את השנה לפניי. סטודנטים
אתה יודע מה יצא? בחודשיים פשוט של המשכורת?
לא.
אבל כלום.
אני חושב שיש שניים שאמרו לי שהם לא יגיעו.
יש אחד שקצת מאחר.
אחד היה לכם איזה משהו בפיזיקה.
זה עניין של מבחן.
יש אחד שלא יגיע ויש עוד מישהו, עוד מישהו של חגי אמר לי שהוא רוצה להגיע. סופרים שניים.
אני לא מה-AT&T.
את לא מה-AT&T? מאיפה את, כן?
אני ממדעי המוח והתנהגות. תואר שני.
אז
למה?
בחוג של
כי
דייר.
אני צריכה נקודות, וחשבתי שאולי ככה אני יכולה להציג ככה.
זה קורסים נקודה
תעבור
בפנים.
ל-AT&T?
מה?
כן.
אוקיי, אז נתנו פנייה עם נקודות.
אולי לא בעיה, אבל...
תישארי לדבר על זה אחר כך, ונראה אם יש פה...
גרוע, אני לא אצטרך לכם.
כן.
אני לא עובדת כמה טובים.
איפה שאנחנו בעברית?
טעות.
Good point. Okay, so welcome everybody to LSF, what used to be the ICNC, and I don't know if you had like probably an official opening. I think I missed it. Anyway, this is a kind of a course that was invented a few years ago. Well, the idea is to in a way keep in touch while you're doing all your separate things which are not still in the program. idea is very simple. We are splitting into a few groups. Each one will have a tutor, one of us, and we're meeting about once a week for 2 hours or so. During the first semester, there's no exam, there's no anything declared officially. We try to get towards some goal because usually it's kind of it makes it easier for everybody. But the goals are very diverse and depending on the instructor and also a lot of our things. So I think what we'll do we want to go? Each one of them will. I think the best is that each one of you will give us like one sentence where they come from. And then we'll present ourselves in, I don't know, 5-10 minutes, what we do and where we come from as well. And then you'll send me an email with your, just 2 preferences, just name 2 people, and we'll try by tomorrow to split you into groups because we're already kind of a week on schedule. And maybe try even this week or next week to make the first meeting. Okay. So let's start.
I'm Amir from Physics and Cognition.
Physics and
Cognition.
Okay.
What's more?
What's your objective studying at the FSU? What brings you to the FSU?
I want to learn more about a lot of things, and I don't know what. I still don't know where I want to make my research. I was last year at the lab, and I think I want to try something different.
I'm also from physics and cognition sciences.
Your name?
Sorry?
Name?
Roy. Yes, I'm not a male, but my sister me and Roy. And any objective I might present would be as a shape. So I want to understand. I want to understand things that are not understood.
My name is Yom. I studied math and statistics. I also don't really have an objective yet, other than trying to learn as much as I can and see what would be what I'm interested in. My name is Omer. I've learned biology and began teaching diploma long ago. I was a teacher for several years, and now I'm doing a master degree in neuroscience and behavior, and another different master degree also in the Hebrew University in science teaching. I'm fascinated by learning. I'm very interested in that. What helps us learn, what does the teacher does or should do. That's where I come from, and I'm very interested in perception as well, visual perception. To be more like that.
One small note, if you're already in a lab, if you're working already with someone.
Yeah, I'm working with Rudolph.
My name is Hagai. I studied biology and physics, and also I don't know exactly where I'm going.
It's a good thing.
Yeah. Everybody says it as if it's a bad thing, but that's.
No, I didn't say it's bad. That's it.
I'm Johannes. I'm here for 1 year only. Mostly I'm working in Eddie Nelson's lab, so I don't know if I can take this tutorial. And I studied maths in Germany and worked in a hospital for 2 years.
You should be. Like the first, are you feeling 50?
more about brain using machine learning algorithm. I work currently at Elon Valley Metropolis at Georgia.
My name is Gal. I studied history and behavioral science, and I'm interested in consciousness, awareness, perception, stuff like that, and everything that relates to it, both from a theoretical point of view and the biological one. And I currently work in Ami's lab, hoping to study the classroom.
Well, Ami suggested we'll go from theory to model. Okay, my name is Yoram, and I'm a physicist in my training. For the past several years, I've been working mostly on questions in neuroscience. Broadly speaking, my interest is on how the brain, or how concrete neural networks in the brain perform various computations. It could be related to memory, it could be related to perception. The topic of the tutorial, as Mickey explained, is very fluid. We can decide on it once we have a group and see what are the interests of everyone. But what I want to try to do during the tutorial is to choose a certain topic, and a certain topic which is interesting theoretically, and use the time to try to understand something about theory. What is it like to develop theory for a certain problem in neuroscience? What can theory do? What are the limitations of theory? It will be nice if we can, depending also on the group, if we can also play a little bit with some modeling, maybe MATLAB, simple things. Of course, we can't do much during the time that we have. And there are a number of topics that we can choose to talk about. I think in the first meeting, we'll just kind of talk broadly about theory. I'll mention several topics, and we'll see what's interesting to everyone. But I can mention one or two topics that I think might be suitable. One is a topic which is close to my heart, and is grid cells. I don't know, did anyone here go to Itzhak Frid's talk, seminar? So I think grid cells are one of the most fascinating discoveries in systems neuroscience, at least in a decade. These are cells that fire when an animal runs around in an environment, and they fire in multiple locations which are arranged in a very geometrical arrangement, in locations that tile the plane, form a lattice, a triangular lattice that tiles the plane. And what we can do is talk about some of the theoretical ideas related to these cells. So Itzhak Frid, in his talk last week, mentioned that these were very recently discovered in humans. They were originally discovered in rodents, in mice and rats. So we can talk about theoretical ideas related to this phenomenon. That's one possibility. There are other possibilities. I think we should just, once we have a group, we can discuss possible ideas.
Okay. I'm Inbal. Like most of you, I also come from several backgrounds. So I have my PhD in both psychology and neurobiology. And then I did my postdoc at Stanford, trying to use both to understand behavior better. I worked there on memory. Here I'm working both on cognitive and emotional functions. And what I learned to do abroad during my postdoc is to use a technique that is now really getting into every wet lab in neuroscience, called autogenetics, in which we use light-sensitive proteins to manipulate the activity of cells in the brain. So I use this technique in order, at the end of the day, to change behavior, but because I have to not just change behavior, but also explain how I'm changing it, it means that in the lab we have to do everything from molecular biology going to happen, but if theoretically it's possible, then it's fine by me. And they came up with great projects that they then presented to. So the idea is to go over the methods and then see how each student can think of the way to use them in a cool way.
Okay, so I'm Miki Lozov. I've been, my undergrad is in computer science, math, and psychology, but I also studied in the program many years ago. And then I did my PhD with Idan Segev on theoretical simulations of individual neurons, also with some aspects in information theory and in collaboration with Tali Dishti. And when I went to postdoc, I switched to experimental neuroscience. And I came back to a lab which is now, I mean, my lab here is now trying to do both experiments. So we're doing electrophysiology and 2-photon imaging mainly, starting now a little bit of optogenetics, trying to in mice. The questions that I'm interested in are, in a way, my feeling is that theoretical, you learn with time about neural networks and the models that we have now. And my gut feeling is that something is missing in these models, and it's something to do with complexity of the single neuron, the fact that neurons have dendrites, and these dendrites are very complicated. So the questions that I'm interested in are mainly on the bridge between the complexity of the single neuron and the coding that the coding of neurons in the brain, about like what in the neural code, what is in the communication between neurons is used by or is affected by complicated properties of single neurons. So to tackle that, we're working on two systems. One of them is in the mice in the bowel cortex. So the mice are actually exploring the environment a lot with their whiskers, and this is a very well-known system in mice. So we can we have a lot of knowledge about the input, about the cortex that is involved in that. So this is one direction that we're working on. And the other direction is actually a system that is very unstudied. It's a very interesting behavior where mice, especially male mice, are emitting a series of ultrasonic vocalizations to female mice, which we're not sure that the female actually hear. It's one question that we're looking at. But this is a very prominent, it's very strong phenomenon. And it's very interesting because there are issues about temporal patterns, about motor control, and especially that it has some similarity to bird song. And what is especially interesting is that we know almost nothing about it. So we don't know where it's in the brain. We don't know if the female hear it, if the male hear it, what controls it, if it's innate or if centrally controlled. So this is like a very exploratory project, but it's interesting to me in the sense that it's brain is like, people are talking a lot about sensory and about motor, and if you want to understand kind of coding schemes, for example, these temporal aspects of coding are important. So many times if you're looking at sensory systems, the time is coming from the sensory stimulus. So you can tell a lot about that. And so you have to look at both aspects of the sensory and the motor if you want to know something general about coding. So that's it. Just about the tutorial, I've done it twice, and both times it went in completely different directions. So it's really up to the students that come in what's interesting, and we can kind of play around with that.
Okay. So I'm Ami essentially. Just I think one thing about the tutorial before I tell you
but to me, I think we probably will have a presentation at the end, right? That was a good
Yeah, we
idea
were
last
trying to
year.
do it last year.
And I think it worked well. So that's
Yeah.
something that you should consider during the semester as we go along, that you don't want to embarrass yourselves too much in front of your friends. So you want to, don't take it as a significant consideration because you do want to go crazy, but at the end we will be coming together and talking and presenting to each other as a group, and we want to learn from that. So it's just at the end, and this is something that could always be done during the last couple of meetings where we coalesce it into something structured and then you present, but just keep it in the back of your minds. So I did my undergraduate here at the Hebrew University, and then went to the Weizmann Institute to study molecular biology in cancer, actually. And then went to Stanford, where I met Inbal when we were coming back to Israel. At Stanford, I worked on molecular neurophysiology, mechanisms of synaptic plasticity, what's thought to underlie learning and memory. And in my lab here, what we do is we marry these two together. We take the tools and the insights that we can gain from studying molecular circuits, gene regulatory networks, understand the response of the brain to an experience, and then we use these tools from molecular neuroscience to study neural circuitry and the organization and plasticity of neural circuitry that underlies behavior. The main model system in the lab is drug addiction, or the response to drugs of abuse. And we start from a behaving mouse and look at its response to drugs of abuse, primarily cocaine. And then we study the gene regulatory networks that are responsive to drugs of abuse, create tools that allow us to identify components of the neural circuitry, manipulate those components of the neural circuitry, and then go back to the animal and ask whether we're modifying the behavior, whether we're modifying the plasticity of the circuit. So basically trying to understand the fundamental components that underlie the encoding of the experience in the mouse, and then perturbing them to see whether we were right in our guess, whether this molecule is important, this connection is important, this nucleus is important for the behavior. Gal mentioned that we're also studying the claustrum, which is sort of a side, something that came out of the mainstream of the cocaine study. The claustrum is a pretty intriguing brain region that has been suggested to underlie consciousness, and we're studying it, and we hope to be able to identify a role for the claustrum in attention and also in the reward circuitry that underlies the response to drugs of abuse. All this time using molecular tools. So I think it's important to recognize that right now we're in the middle of a pretty dramatic revolution in neuroscience, where up till now there was a separation between classic neurophysiologists and molecular neuroscientists, and now the two fields are coming together and creating a synergy where molecular tools are being implemented in the study of neural circuits, allowing us to recognize circuitry in high resolution, more detail, be able to identify the function of the circuit and its plasticity, and this will also be the topic of my group in the tutorial. So what we'll do is we'll look at the variety of tools that are available and how to implement them, and we'll do this what we did last year. Hopefully we'll improve on what we did last year and put it together in a way that our objective would be to create a model system. Each one of you will come up with, or the people that choose to join the group, will come up with a question that's intriguing for them. These could be crazy questions. What is consciousness? Where does it lie? How does the brain compute value? Where are different modalities integrated in the brain? Can we expand our capacity for memory? Like these fundamental issues in neuroscience, and then during the semester we'll try to create an experimental system that allows us to tackle this specific question using molecular tools. And, in parallel, what we'll do is we'll expand and improve our skills for critical reading of scientific literature on cutting-edge papers that are published weekly now in top journals, using molecular tools to understand how the circuitry is organized. So we'll critically read those and then be able to get a better understanding for the tools that are out there, and then see how those can be implemented in the crazy project that you'll be developing. What else? Yeah, I think that covers what. And obviously there is a lot of flexibility within each of these. We can wander. It's a path that we initially try to focus, and then we wander around where the interest of the group takes us. Okay. So I think you have a pretty good overview. Probably half of the things we said, I'm not telling you anything, but you kind of have at least a clear view of what we sound like. And I think the best is that if you, so just one thing I thought would be, we have to say, we are a non-representative group of the people in ELSA. So we are the kind of young generation, so to say. It's very difficult to be a young scientist today, because by the time you get a job, you're already not too. You don't need to demoralize. But you will be better, of course. Yeah, you guys will maintain your good looks and your youth by the time you. There are techniques of coming up to save your, especially molecular. But what I was trying to say is that we are not. First of all, we're not spanning all the areas. So there are people who are from psychology that are not represented, from medical, from other computer science that are not kind of here. So this is not. You shouldn't be kind of intimidated by the limited choice you have for your PhD. So it might be that your interests, although they seem to be very broad and untuned, even if it's not exactly what you were thinking, I'm sure you'll enjoy this tutorial. Practically, just send me, and try to do it today, an email with 2 names that you prefer to be. We can't promise anything. It might turn out that everybody wants to work with one person, and then we'll have to do something about it. But it should work. And in any case, I think it will be great. So send me an email, and we'll try by tomorrow to send you an email back, and then we'll schedule the meeting.
Can you write down your email address?
Yes, sure.
Yeah, while Mickey is writing, sorry, I asked Ruti to ask you, there was a paper in Neuron that was completely non-scientific last week. Did she send it to you?
Yeah.
Achla. Read it. This week you'll still have time. Next week you really start studying for it.
So it's Mikilon, MIKILON @gmail.
And when do we want by today in the afternoon, or today, anytime today?
Yeah, make sure that today by midnight you have the email back, okay? Just have deadline. We missed somebody. You didn't have a chance to introduce yourself.
My name is Dan. Sorry that I can play.
You're working with Odai.
Right.
Right, okay.
So
everybody said what their background is and what
I'm an electrical engineer. My background with the BS and MS.
And you're working with Chagai now. say you want to identify a group of cells that is involved in behavior. So how would you know what group of cells you're looking for? You need what makes cells unique, what makes them function, and what makes them unique are the proteins that they express, the repertoire of genes that they utilize. So in order to identify subsets of neurons that underlie a particular behavior, you need to be able to identify the genes that they utilize, and then you can use these genes to create a growing variety of tools, including optogenetic tools that you can target to this specific population in order to either study them in high resolution, so study their gene expression in a particularly their gene expression, or to manipulate their activity, or study their connectivity, what neurons interact with them and where they send their processes to. All this you can do using implementing a variety of molecular tools once you can identify a population.
So it's not really very specific thing. Molecular tool is just a broad name for all these different things.
It covers a large spectrum, yes. It covers all these tools that you can implement in a specific context.
What are the challenges using the optogenetics and what you expect right now with the monkeys and not with mice?
So you're asking about optogenetics in monkeys?
Yeah, why it's very challenging?
Why what?
Why it's challenging, why it's used in mice and not in other...
I can tell you my honest opinion, which is that monkey people are way more conservative than mice people, and for a good reason. I mean, monkeys are way more expensive in many different ways, so people hesitate more before. If someone now wants to integrate optogenetics into a mouse lab, that you just buy the virus, inject it, and see what happens. To do that in a monkey, just finishing the thought process would take forever.
But also,
so in the last, that I worked with, people did work with monkeys, and there was a paper published last week, I think, Shai's paper. I think it was Shai's paper, yeah. Last week. Well, when you're exciting, or you think you're exciting, neurons electrically, you're actually doing something that's very unspecific, right? You just injected an electrical current, which activates excitatory neurons, but also inhibitory neurons, and all the glia cells around it. And the bottom line is you have no idea what you're doing, right, when you're using electrical stimulation. The idea with optogenetics is to specifically target just neurons, or just excitatory neurons, or just a subpopulation of some kind of neuromodulatory group. And then, by definition, the effect that you'll get will be lower. So, for example, the paper that came out last week combined electrical stimulation and optogenetic stimulation, and the problem is always to get an effect on behavior. So there are some mild behavioral effects already achieved with monkeys. It's getting there. It's more difficult to target a bigger area with a virus, and there are some technical difficulties as well. I can explain everything.
And also, aside from that, you don't have the genetic, we're talking about the genetic tools.
No, the promoters work just fine.
No, but you
in
can't
monkeys.
match as well as you can in mice.
You don't have screen monkeys also, yes,
yeah.
but no, but the regular promoters, the cancave or excitatory, and no, it works just fine in monkeys.
But maybe just, there are more people. Mice have been traditionally a genetic model organism, so there are people who are used to work with mice, many more people who are used to work with mice and use these kinds of tools than people who work with monkeys.
Yeah, there are several labs worldwide now that are working with monkeys, but for example, the person that I know that works with monkeys moved to rats. She has a monkey PhD, and she did a monkey course, and now she has because rodents are more flexible.
What about time-wise? Don't you think optogenetics takes more time in monkeys because their growth is quicker?
לעבור להרביץ.
לא, כאילו, לכאורה היא אמורה להגיד שזה כן.
אני לא הייתי
מה שכן, זה כיף לי
טוב, מה, יש לנו... לא, רצינו, זה לא הייתי לא, 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 לא. נראה לי יותר הרבה טובות. נראה לי מהר שפו מישהו לזה.
בסביבה אחרת הוא היה מציין את זה, לפעמים כזה, תראה את זה, כן. גדול הוא עשה שנה, אבל לא יודע מה נתוני שזה,
יכול להיות שתשאר נושאים של סגנון אחר מאוד של תיאוריה מה...
נראה לי, בקיצור אתם פשוט הייתי שמה.
ואני רוצה להכיר דיפרל, הייתם הרצאה ביום חמישי?
ביום חמישי של...
סיפר שם על כל השאר דיפרל, והמראה שאחרת באיזשהו כאילו מה וירטואלית, יש להם שיעורים
שמרים להם תמונות?
יש להם שיעורים מסודרים בצורה גיאומטרית, האמת שזה לא היה כל כך ברור בהרצאה שלו, תקליט שפרקשה בחרפנים,
גריפשן זה בעצם הרי של אלקטרודות?
לא, עכשיו אבל מזריקים את הסכולים הזה,
לא חומר ניגול. לא, אבל זה מולקולות, זה כמו ג'יקאם. ג'יקאם ל-FMRI. האמת שאני התחלתי פרויקט הזה עם מישהו אצלנו במעבדה לפני זה.
נודדים תאים באזור של גן תורן הקורטקס.
צריכים לעשות סיפור דיפנדנט.
אני לא נתן.
אתה יודע מה?
עשיתי כמה קורסים פיזיולוגיים,
לא, אז לא היה כלום על זה.
אולי אתה יודע שיש תאים שנקראים פלייסר?
זה לא בדיוק ככה, אבל מה שהכי נהרג אבל המאמר יצא לפני, הוא התחיל את הרעיון ביחד עם חיים והמאמר
שלנו מעניין הרבה תאים בהיפוקאמפוס, הוא מודד שאומר שהם יורים באזור מסוים. מה שגילו לפני שבע שנים זה
קונטרסטיישן של המראי, משהו שתופס את המחור הזה, ואז אתה יכול לראות פעילות נראים פעם במחשב.
רן, אתה הכרת את רן?
אוקיי, בעצם תא מסוים, זאת אומרת כל פעם זה תא אחר שירה? אותו תא, אבל רק בנקודות מסוימות.
עכשיו הוא עובד
סוג
של GPS במרחבים.
יש הרבה מאוד שאלות, מה התפקיד של זה? איך בכלל נוצר הדבר הזה?
אז הוא עשה,
זה נושא שעבדתי עליו.
הוא עשה, זה לפתע אני חושבת היה, מרקר וזה בחור ישראלי, כאילו, המנהל מעבדה, או יוסי, זה
יש מישהו בסטנפורד שגם עושה כל מיני קליפות, דברים שהם מקליפים בין מיני קליפות ואז משחררים איזשהו, וגם האמת, אם אתה באמת רוצה לעשות את זה, יש לו, בהדסה יש מישהו שעשה פוסט-טיפות, ועושה דברים ממש מדליקים, לטורינס בעיקר, לראות ממש דברים יפים, מאוד מאוד מדליקים. אני חשבתי לדבר איתם, לא, אבל זה כאילו חופש ופיתוחים מולקולריים.
אביב, הרקע שלו בביולוגיה וכימיה.
אבל הוא בא בזה?
כן. זה בעיקר פעמים כאילו, אם הצליחו לעזור לו מה העניינים אותי? כן, אני עדיין